All right, hi, my little bio students. This video is going to be on pages 9 and 10 in the notes, okay? And this is basically finishing out anaerobic respiration, okay? So we talked in the last video about how anaerobic respiration begins, and that is with glycolysis. So just as a quick review, during glycolysis, we took a glucose and ended up splitting it into a few different things. We used 2-ATP in order for activation to occur. And at the end of glycolysis, we got a total of 4 ATPs, but a net of 2. And if you don't get that, make sure you go back and look at the other video. We had 2 pyruvates, and we got 2 NADH2s. Okay, so that's a quick review for glycolysis. Now, at this point, this is where either aerobic or anaerobic respiration is going to occur once we have those pyruvates. If we have oxygen present, you're going to have aerobic respiration. If you don't have oxygen present, you're just going to finish out with anaerobic respiration. Okay, so that finishing process, if no oxygen is present, that's called fermentation. Okay, and basically with fermentation, you end up during glycolysis, we produced these NADH2s, okay? Problem is, if oxygen is present, eventually the NAD can drop off the hydrogens here to the oxygen, okay? So that's if it is present. But if it's not present, these NADH2s have to be oxidized and get rid of these hydrogen so that they can go back and pick up more to continue with glycolysis. So fermentation is basically you actually don't produce any more ATP. Okay, fermentation is just a way to drop off these hydrogen so that then NAD can go back and pick up more hydrogen during glycolysis. Because if all the NAD get full with hydrogen and can't drop them off, eventually glycolysis is going to stop because there's no way to pick up those hydrogen. Okay, so fermentation pyruvate becomes the final hydrogen acceptor. So basically what happens is during glycolysis you created pyruvate, and then during fermentation, there's 2 options, but basically that pyruvate is going to accept back those hydrogen to free up the NAD to go pick up more. Okay, so it says in anaerobic respiration, all of the energy obtained from the cell comes from the process of glycolysis. Okay, so no more energy during fermentation, no more ATP are produced. After glycolysis has occurred, the pyruvic acid or pyruvate is changed to other compounds and products with no further release of energy. This is known as fermentation. This occurs in the cell cytoplasm. Okay, so after glycolysis, pyruvic acid may be converted into, there's 2 options, and depending on the organism. Yeast are an organism that they do what's called alcoholic fermentation. Okay, that is this, where basically you take your pyruvic acid and NADH2 comes in and drops off those hydrogen to become NAD, which can then go back and pick up more hydrogen. And when it drops off those hydrogen, what happens is a CO2 comes off, and then you also get a ethyl alcohol. Okay, so that's basically it then. So you get ethyl alcohol, you get a CO2, and the big thing is your NAD can now go back and continue with glycolysis. Okay, because if not, they all get used up. Or in us and most animals, what you can do in anaerobic respiration is the pyruvic acid or pyruvate accepts these hydrogen, and what you form is lactic acid. Okay, so 2 options, and it's not like a cell can do either one. Some cells, such as yeast, do alcoholic, other cells do lactic acid, but they both they're basically the same general process. So you can see on your notes the first structural equation that you have there. You take pyruvic acid, NADH2 comes in, the hydrogen come off, the hydrogen get added then to the pyruvate, and that causes it to split apart, and you release carbon dioxide and you get ethyl alcohol. Okay, and if you count the hydrogens on pyruvic acid, you have 1, 2, 3, 4. If you count the hydrogens on the alcohol, you have 1, 2, 3, 4, 5, 6. So it basically picked up those two hydrogen. Okay, alcoholic fermentation, this is how obviously we make alcohol, but it's also yeast. That's why bread rises, because of the CO2 that comes off. That causes air pockets in the bread, and that's what makes bread rise. That's why you add yeast to bread when you cook it. You do get alcohol too, but that burns off when you cook it. Okay. Now in humans, what happens is same general idea. NH2 comes in, drops off the hydrogen, but instead of CO2 splitting off, you just get lactic acid. Okay, and you can kind of see your notes there what lactic acid looks like. And if you count the hydrogens again, what happens is the hydrogens get accepted by the pyruvate, and that turns into lactic acid. Now lactic acid, that's going to, when we produce that's actually a toxin, and that makes your muscles really sore. You don't want to do a lot of anaerobic respiration. You don't want to do a lot of fermentation. You can do it if needed, if you're doing something strenuous, but you actually don't want to do a ton of that. So it makes your muscles really tired, which is basically a way of homeostasis being maintained, because if your muscles are really tired, you're going to eventually slow down and stop doing anaerobic respiration. Okay? So basically you can see fermentation definition. So underneath those equations, it says definition: after glycolysis, the breakdown of pyruvic acid with no release of energy. So basically, pyruvate becomes the final hydrogen acceptor, and you don't get any more ATP. Okay, so now anaerobic respiration is complete. Okay, so summary of anaerobic respiration: oxygen is not required and does not participate in the biochemical sequence. Begins with glycolysis and ends with fermentation. It occurs in the cell cytoplasm, and as far as ATPs go, you use 2 as activation energy. You form 4 from glycolysis. You have a net of 2. Okay, you don't form any from fermentation. So the whole purpose of fermentation is just getting back your NADs to go back and pick up more hydrogen. So you get a net gain of only 2 ATPs. Okay, and this doesn't sound like a lot, but for small cells like bacteria and yeast, this is enough to survive. Not enough to survive for most multicellular organisms, but for small cells, this is enough. Okay, so if you flip over to page 10, you can see your end products for anaerobic respiration. In glycolysis, your end products are pyruvate. I would add in there pyruvate, I would say 4 ATP, I would say net of 2, and I would also say you get NADH2, 2 of them. Okay. In alcoholic fermentation, you get ethyl alcohol, you get carbon dioxide, and I would add in there, you also get NAD, because the hydrogen gets dropped off, and that's what causes this change to occur. And in lactic acid fermentation, I would you get lactic acid, and I would also add you get NAD. Okay, so your summary equations you can kind of see there. That first one there, that's glycolysis. We talked about that yesterday. You take glucose first, you break it down into pGal, then you take that pGal and you oxidize it into pyruvate, and then during alcoholic fermentation, I just took a picture of it, and now I added a few things, so make sure you do this. For alcoholic fermentation, you can see that you take your glucose, you use your glucose, and you use 2 ATP. This part is glycolysis up to here. Okay, this is what you get from glycolysis. I would add in you get 2 NADH2s. Sometimes that's not shown because it's a coenzyme, but for our intents and purposes, I would show it because it's important. So I would add plus 2 NADH2s. Then what you get during fermentation, so to go from here to here, this is fermentation, and you get ethyl alcohol, carbon dioxide, and you get NAD. And then this NAD is going to go back and pick up hydrogen during glycolysis. Okay, so then it can just be recycled. So fermentation is a way to recycle that NAD. Okay, lactic acid fermentation is the same general thing. This part here, glucose plus 2 ATP, this is glycolysis. And then during glycolysis, I would also, again, I would add, you get 2 NADH2s, and then the NADH2s drop off the hydrogen to the pyruvate, and what you get is 2 lactic acids, but then you also get those 2 NADs that form, and they're going to go back and continue glycolysis. So this diagram here just kind of shows that. Basically, it's kind of hard to tell which way the arrow is pointing, but the arrow is pointing, and you might want to kind of fill this in, the arrow is going this direction. Okay, so during glycolysis, NAD picks up the hydrogen. During fermentation, NADH2 drops off the hydrogen, and you just get this cycle. So this is a way to reuse the NAD, because it has to be oxidized somewhere. Okay, so that is your, that's basically, we're done with anaerobic respiration. Okay, so you start with glycolysis, then you do fermentation as a way to regain your free NAD molecules. Then the last thing we have to talk about, we're down to just 3 pages of notes, 11, 12, 13, probably 2 or 3 videos though on that, and that will finish up with aerobic respiration, which is a little bit different. In that case, since you do have oxygen, pyruvate's going to go through a few more things and get broken down, and then eventually what accepts the hydrogen from all the NADs and FADs, we'll learn about in a little bit, but that's going to be your oxygen. Okay, so a little bit different. But that's your, I think this is up to video, I want to say it's like video number 9, and that was on pages 9 and 10. All right, so peace out, and stay tuned for the next few videos.